Hello students! For today we start talking about other topic called bioenergetics. First, let's think about metabolic classification of organisms. The nutritional requirements of an organism reflect its source of metabolic energy. There are different types of organisms. The first types are called autotrophs. They synthesize their cellular components from simple molecules like water, carbon dioxide, ammonium, or hydrogen sulfide. The other types of organisms are called chemolithotrophs, which obtain energy by oxidizing inorganic compounds. The next ones are called photoautotrophs, which use photosynthesis like all plants that you know. The light energy drives the transfer of electrons from inorganic donors to carbon dioxide for the synthesis of carbohydrates, and all the rest, or we, are heterotrophs, which obtain energy from complex compounds called carbohydrates, lipids, and proteins to form energy in the end. Ultimately, we depend on autotrophs. The organisms, according to the using of oxygen or not, are classified of number one, obligate aerobes, or animals which obligatory use oxygen. Actually, we are obligate aerobes too. Anaerobes are these ones that don't use oxygen. They use other oxidizers such as sulfate or nitrate. There are two types: facultative anaerobes like Escherichia coli, which can grow both in presence or absence of oxygen, and obligate anaerobes like Clostridium difficile, which need absolutely the presence of oxygen. Animals and humans are obligate aerobic heterotrophs whose food depends on the balanced intake of macronutrients, which are carbohydrates, proteins, and lipids, and micronutrients, which are vitamins and minerals that we talked about. Humans are among the most complex organisms on Earth and require a continuous supply of energy to maintain their structure and functions. Actually, students, the primary source of energy for most living organisms is the sun. We need the sunlight because either directly from the plants or indirectly thanks to the animals we can take this energy from the sun, because we cannot photosynthesize. That's why we can take this energy only thanks to the plants. And humans obtain energy through plants and animal food containing carbohydrates, fats, and proteins, which are broken down to form ATP, and this is the so-called energy currency for the cell. ATP, you know, means adenosine triphosphate. This molecule is energy for us. There are many types of chemical energy in the human body. The forms are light energy, electrical energy, mechanical energy, chemical that we use, thermal in biology. Actually, this chemical energy is the most important source. Chemical energy in nutrients. We take energy from nutrients, which is transformed into ATP, which is the universal energy carrier. Why we need energy? Why we need this energy from the food actually? For the first, we need energy because of the biosynthetic processes in our organism, students. Actually, all molecules in our organism have some period of life. After that, they will be destroyed. And we need to regenerate all these structures. For that reason, all the time we need biosynthetic processes to replace the older compounds that are destroyed before that. What are these biosynthetic processes? Synthesis of new proteins, new carbohydrates, new fats, new hormones, new receptors, or etc. Number 2, we need energy for muscle contraction. Why? Because we need our muscle for the moving of our body. Number 3, for the generation and conduction of action potentials in the nerves, in the brain. Then, for the active transport through the membranes. Actually, the cell produce or can take different compounds from outside the cell that need to be transported into the cell. For that reason, we need to have intact cell membranes, and many times the transport requires energy, requires ATP. That's why this type of transport is called active transport. The next one is for the maintenance of structure and organization of cell organelles, of storage and transmission of genetic information, and for the maintenance of body temperature. Energy and cellular processes. The Gibbs free energy, delta G, means how much energy from a process can be used to perform work at constant temperature and pressure. During the catabolic processes, which means degradation of polymers to monomers, there is association with decreasing of Gibbs free energy, which means delta G in catabolic processes is less than 0, and these processes occur spontaneously, and during these processes energy is released. That's why the other name of these processes is exergonic. The next type of opposite processes, which means synthesis from monomers to polymers in our body, from simple compounds to complex compounds, are called anabolic processes. In this, delta G or Gibbs free energy is more than 0, which means these processes require energy. They need energy, usually from ATP or other compounds with that are energy carriers. Direct coupling of all these opposite processes, catabolic and anabolic, is impossible. Absolutely impossible. That's why there is indirect coupling by such of these energy intermediates like ATP, GTP, NADH, NADPH, FAGH, acetyl coenzyme A, and etc. The general laws of thermodynamics state that the first law is energy is neither lost nor created, only transformed, or there is conservation of energy, there is saving of energy. This means that energy is not loose in our universe, it is constant, just transforming of energy there is. The second law of thermodynamics states that in an isolated system, entropy, the other word of entropy is a disorder, increases, and spontaneous processes proceed in this direction with increasing of entropy or with increasing of the disorder. The free energy determines whether a biochemical reaction occurs spontaneously and can perform work. Actually, students, you know the 6 main groups of enzymatic reactions. Do you remember transferases, ligases, lyases, isomerases, and etc. But only one type of reaction can be used for energy production, and these are the type number 1. These are oxidoreductive reactions. Only these reactions can be used for the production of energy. Why? Because of the electron transfer. That's why. Living organisms are open system. Actually, they exchange matter and energy with their surroundings. Therefore, when applying the laws of thermodynamics to biological systems, it must be considered that the organism is not isolated, but is part of the larger system, organism plus environment. Why? Because, students, we need different complex molecules which come from outside, from the environment, and release outside different end products like carbon dioxide and water as urine, and during this time heat production there is in the end. The first law of thermodynamic, conservation of energy, and the second law, increase of entropy, apply to the entire system, organism plus environment, not only for the organism. This allows the organism to maintain low entropy inside by increasing entropy in the environment. You see, what is this? Free energy and spontaneity of biochemical reactions. The standard free energy change means this is the theoretical constant for a given reaction under standard conditions, which means pH around 7, temperature around 25 degrees Celsius, and 1 mole concentration of all reactants and products. And the spontaneity of the reaction depends on delta G, the changes in the free energy. The fundamental formula is this one: delta G is equal to delta H, which is change in enthalpy. Enthalpy means the total heat energy change of a system at constant pressure minus temperature in Kelvin is multiplied delta S, which is the changing in the entropy or changing in the disorder of the system. And if the reaction is spontaneous, delta G must be less than 0. Less than 0. This is the spontaneous exergonic reaction. But this means that if delta G is less than 0, it depends not only from the entropy, which is delta S, it depends from the total heat energy of the reaction also. For example, if entropy decreases, which means delta S is less than 0, the reaction can still be spontaneous if delta H is negative. Only in this situation, negative delta H means exothermic reaction. To summarize, what are these exergonic and endergonic reactions? Exergonic reaction or spontaneous reaction means delta G is less than 0. Exergonic reaction, this is the exergonic. Endergonic reaction means when delta G is more than 0, or positive will be the change of Gibbs energy, and can occur only with an input of energy. When delta G is very, very low, the reaction proceeds almost completely to the end and is effectively irreversible. And when delta G is equal to 0, the system is on equilibrium with no net change occurring. I will give you one example to understand, students, what is this increasing or decreasing of the disorder. You can see this picture before that. It's one cyclist that goes down the hill. When he goes down the hill, this is actually the spontaneous process without any energy. Actually, no require energy during this spontaneous process. This spontaneous process is the same when we talk about the exergonic catabolic reaction in our body, and in this situation delta G will be negative because this reaction is spontaneous. But when the cyclist goes up the hill, this means that he requires energy. He requires more energy to go up the hill. In this situation, delta G will be positive. And this picture is the same like anabolic reactions in our body when we build up the compounds. That's why this is the same like catabolism, which means breaking down the compounds, and this is like anabolism when where means synthesis of the compounds. One more example I can give you. Imagine that you have one chessboard on the table. You walk and bumped and push the chessboard and it falls the ground, and all pieces are scattered. After that, and this is absolutely spontaneous process, this process shows that delta G is absolutely negative. This process leads to the increasing of the disorder, increasing of the chaos, of the randomness of the system. And one more, during this falling down of pieces, you can hear a noise, which means energy is released during this time. Imagine the opposite process. You need to arrange this chessboard in order. You need to pick up one by one all pieces and place them in the corresponding position in the chessboard. You need energy to do that. This means delta G in this situation is positive. It's like the same picture. What will be happen about the disorder? The disorder will be decreased. The chaos, the randomness will be decreased. And the same processes we have in our body, but because we are organisms that don't eat all the time, 24 from 24, we eat during some period of time. That's why we have specific names of this period that we eat: breakfast, lunch, dinner, and etc. Between these periods of eating time, these periods are called periods of starvation, of the fasting state. And that's why, students, but we need energy all the time, not only during the eating times. We need energy from 24 hours all the time. That's why exist some compounds that are called high energy compounds. This compound, the other name is macroergic compounds. We need them because of the saving of energy. That's why, students, bioenergetics studies not only how to produce energy, but how to save and how to use. And thanks to these high energy compounds like ATP, you know one of them, ATP, we can save energy, and we can use these compounds during the times that we are in the starvation states. What are these normo and macroergic compounds? Normoergic compounds means that they contain normoergic bonds. This means bonds with normal energy after their hydrolysis, which means that the standard free energy is changed upon the hydrolysis with releasing of 8 to 21 kilojoule per mole energy. As examples: ester bonds, glycosidic bonds, peptide bonds. Macroergic bonds or high energy bonds are these ones that after their hydrolysis more than 30 kilojoule per mole energy is released. They are called high energy bonds, and one of them is ATP, and this bond is represented by this symbol, this one, like wave. This means high energy bond. This table shows all high energy bonds in our body. Let's see one by one. The first one, the richest of energy compounds in the first one, which is called phosphoenolpyruvate. Whereas, look here, this is the macroergic bond, which is called NO phosphate bond. And after the hydrolysis of this bond, 61.9 kilojoule per mole energy is released, or delta G is less than 0, there is minus before that. What is the role of this compound? This compound is used in the processes of glycolysis and gluconeogenesis. You'll see when we talk about these processes. The second one is called 1,3-biphosphoglycerate, which has so-called acyl phosphate macroergic bond. Again, in the glycolysis and gluconeogenesis is used. The third one is called creatine phosphate. By the way, you know something about this compound. Do you know some information more about creatine phosphate? We said something before. Actually, you know the enzyme that produces this compound. Enzyme name was creatine phosphokinase, which has 3 enzymes, BB, and MB. And the MB isoenzyme was a very important indicator for myocardial infarction. Do you remember? Yes. And the bond here is called guanidine phosphate macroergic bond. This compound is used as a reserve macroergic compound mainly in the muscle tissue, but not only. Can be found in the brain and in the heart too. And various acyl coenzyme A, and for example acetyl coenzyme A, which have thioester macroergic bond too. Let's think about the differences between acyl and acetyl coenzyme A. Do you know that there is very big difference? What is this acyl coenzyme A? The first compound. Acyl means any fatty acid plus coenzyme A. This is the acyl. Acetyl coenzyme A means this compound, this one, CH3CO-CoA. This is the acetyl coenzyme A with 2 carbon atoms. But in both of the types of the compounds, there is thioester macroergic bond. And the last ones are ATP macroergic bonds, which are beta and gamma. Do you remember that there were 2 macroergic bonds, and after their hydrolysis can be produced a huge amount of energy if there is full hydrolysis of both of the bonds, not only the one. And this is the structure of ATP. I will remind you that this is the base, adenine, this is the pentose, and after that there is first, second and third phosphate atom. This is called the alpha phosphate atom, and the bond is normoergic. This is called beta phosphate atom, and the bond is phosphoanhydrate bond. It is the high energy. And the last one is called gamma phosphate atom. Again, the bond is phosphoanhydrate and is high energy too. Here, students, we can remind one more how many types of hydrolysis there is in the molecule of ATP. Do you remember? Tell me, if there is hydrolysis of beta bond, which will be the product? Beta bond hydrolysis. Tell me the product. Beta. The product first one will be AMP, yes, plus 2 phosphate atoms, which are called pyrophosphate. But into the pyrophosphate there is gamma macroergic bond. After the hydrolysis of this gamma by pyrophosphatase enzyme, again energy will be released. Tell me the product after the hydrolysis of gamma bond. ADP plus P, yes, plus 1 inorganic phosphate, yes. And to summarize, it's very important, students, be focused, be concentrated, because this is the most important part of all metabolic processes. You need to know all this in general, and after that, specifically one by one in all processes. We have metabolism in our body, which consists of 2 opposite processes. The first ones are called catabolic processes, which means processes of breakdown of macromolecules. Which are they? Carbohydrates to monosaccharides, proteins to alpha amino acids, and lipids, mainly triacylglycerols, to fatty acids and glycerol. These processes are called exergonic because delta G is less than 0. These are spontaneous processes that produce energy. That's why if I ask you what is the role of this process, first you need to ask yourself if this process is catabolic or anabolic. If the answer is this process is catabolic, Your answer have to be, the role of this process is energy production. Examples. Glycolysis. What is the role of glycolysis, students? Just repeat. Energy production. What is the role of beta oxidation, students? Energy production. What is the role of respiratory chain? Why? Because all these processes are catabolic. That's why. One more. I said that the reactions that are used for the production of energy are oxidoreductive reactions. That's why we need to clarify what kind of processes are catabolic, oxidative or reductive ones. Remember, students, these processes are processes of oxidation, which means dehydrogenation. We need to take hydrogen atoms from polymers for something. You'll see why. For all these dehydrogenative reactions, we need two main cofactors. These are NAD or FAD. These are primarily the cofactors. And one more. Remember one more, students. These processes are active during the fasting, during the starvation state. That's why at this moment we don't have insulin. We have glucagon, adrenaline, and all contrainsular hormones, but not insulin. Let's see all the rest. The opposite ones are called anabolic processes. These processes are endergonic, which means delta G is more than 0. They require energy. They need ATP. Without ATP, these processes are impossible. What is the mechanism? They are used for the synthesis from simple molecules, complex molecules. Example, from monosaccharides, carbohydrates, from amino acids, proteins, from fatty acids and glycerol, triacylglycerols, or many other examples there are. And according to the oxidoreduction, these are opposite of oxidation, which means these processes are processes of reduction. This is the opposite process of dehydrogenation, reduction. But in this situation, the cofactor is NADPH. No NADH, no FADH, NADPH is used as a cofactor of all reductases, and the enzymes are called reductases. And the examples are all biosynthetic processes, muscle work, nerve excitation, cell division, and etc. When are they active? To start synthetic processes, we need to have enough substrates. Enough substrates, which means enough food. They are active during the well-fed state. That's why during this time we have opposite hormone of glucagon, insulin. Remember that. If you hear well-fed state, insulin. If you hear starvation state, glucagon. And the coupling of both of the processes is thanks to macroergic compounds like ATP and some others. This is the same. On the left are energy release processes like glycolysis, beta oxidation, and etc. Then energy is produced in the form of macroergic compounds. Then energy is used for the reductive, for the synthetic, endergonic reactions. This coupling of both of the processes you can see in this example of the phosphorylation of glucose. You know this reaction from the previous lectures when we talked about one enzyme that is used for the phosphorylation of glucose. Do you remember the name? From glucose to glucose 6-phosphate. This is the reaction. Do you remember the enzyme? What was the enzyme name of these enzymes that phosphorylate thanks to ATP? What was the enzyme name? Yes?
Protein kinase.
Yes, well done. They are called kinases. This enzyme is hexokinase, and it is used in the first reaction of glycolysis process. Actually, there are two reactions that are coupled. The first one is glucose plus inorganic phosphate, and glucose 6-phosphate is produced. The reaction is non-spontaneous because delta G is more than 0. The reaction is endergonic. And the next one, which is coupled with the first one, is hydrolysis of the gamma phosphate group of ATP. And this phosphate goes here, actually. This phosphate is used here. But this reaction is possible. This is strongly exergonic reaction, spontaneous, which leads to energy production because delta G is less than 0. That's why the coupled reaction will be like this. In the end, the reaction will be happened. This scheme, this figure shows this connection between catabolic exergonic reactions that are used for the degradation of complex compounds to simple ones, and during this process ATP is produced from ADP plus P. These are the substrates. Then these simple molecules enter the anaerobic processes to form again the complex ones, and heat production, the dissipation there is during the same time. Students, let's talk about ATP. ATP is not the highest energy compound actually. In bioenergetics, cannot be transfer of energy directly from the richest to the poorest. Cannot be directly, no. That's why we need one intermediate which is called ATP. There is transfer of energy from the richest to the ATP, from the ATP to the poorest. Understand? You see now in this example, this creatine phosphate, we talked about this in the table before that, was one of the examples of high energy compounds with guanidine phosphate macroergic bond. With the very rich of energy compounds. But after the process of transfer of this macroergic phosphate to ADP, ADP is an acceptor. ADP will be converted to ATP and creatine will be produced in the end as a product. But we have ATP now, and ATP can be used after that in the next processes. Look this scheme. On the left are all high energy compounds like phosphenol pyruvate, 1,3-bphosphoglycerate, creatine phosphate, and etc. They transfer their phosphate macroergic bond to ADP to form ATP. After that, ATP is used for all these phosphorylation reactions to give energy to the compound with low energy. The richest of energy compounds, the highest of energy compounds, the lowest of energy compounds. But thanks to ATP, without ATP, these processes will be impossible. Now, ATP plus glycerol, glycerol 3-phosphate will be produced. Glycerol 3-phosphate has more energy than glycerol before that. Other reaction that we said before. ATP gives energy to glucose, and glucose 6-phosphate is produced, which means the energy will be increased in the glucose 6-phosphate more than before that the glucose. Biological oxidation. What is this biological oxidation? Actually, students, with simple words, this means energy production in the body. But before that, we need to explain what is this oxidation, what was this reduction. Do you remember? What was? Do you remember, students? The processes of oxidation is releasing of energy. The electrons are lost. And that's why the compound A is oxidized. The process of reduction means that this compound will accept hydrogen atoms. That's why it will be reduced. For example, Nag plus is oxidized form, NaGH is reduced form. And the next. We said about changes in the free and Gibbs energy, but when we're talking about the body, the human body, the living organisms, we need to talk about one more indicator, which is called redox potential. Redox potential, and this is the difference in the standard redox potential between electron acceptor and electron donor. This is the indicator which is called delta E. Delta E. This relationship between these changes in the standard free energy need one more factor, one more indicator, which is changing in this standard redox potential. What does it mean? This means that all the time we'll talk about transfer of electrons between donor, gives electrons, acceptor, accept electrons. Again, gives electrons, accept electrons. But a redox potential, students, means the affinity to the electrons. Compounds with low redox potential means that they have low affinity to the electrons, which means they will accept the electrons but will give rapidly. But the compounds with higher redox potential, which means that they will accept the electrons with more affinity. Understand? And in the end, the final acceptor of electrons will has the highest affinity to the electrons. From this table, you can see which is the compound with highest affinity to electrons. This is oxygen. Oxygen is the compound with highest affinity. And in this table there are many compounds with different redox potential, like cytochromes. They have more affinity than ubiquinone, fumarate, FAD, oxaloacetate, pyruvate, lipoic acid, NAD, and etc. These are some basic principles of biological oxidation. We said about oxidation and reduction, what is this? And remember that the electron transport is from redox systems with lower redox potential to redox systems with higher redox potential. But one more, what is this redox system? Let's explain what is this. Do you know what is this redox system? No. Okay, redox system, actually we said many times about redox system in the previous lectures. Redox system means deoxidized and reduced form of one compound. NAD, oxidized. NADH, reduced. FAD, oxidized. FADH, reduced, and etc. You'll see today. According to the environment and to the final acceptor of electrons, we have two types of cells. Aerobic cells, where the terminal acceptor of electrons is oxygen, and anaerobic cells, where the final acceptor of electrons is not oxygen, is something else. That's why there are two chains, aerobic chains with oxygen as the terminal acceptor, and anaerobic metabolic chains with another acceptor which is not oxygen, maybe NAD or maybe FAD or something else, but not oxygen. And the students, three stages of aerobic metabolism we have, three phases. The first phase is the phase of digestion or degradation from polymers to monomers. No oxidative reactions here. The second is formation of acetyl coenzyme A. You will see now. And the third one is Krebs cycle and a respiratory chain. And don't be scared. This is the scheme which shows the aerobic processes. Students, actually the first phase is this one, the first line. This is the first phase of so-called digestion, but it's not digestion called during the in the organism. With yellow color, this is the second phase, and all this with pink color and green color is the third phase of metabolism. Actually, it's not only scheme, it's a map with all processes, almost all, because no many places here, but you need to draw this scheme at your home. You can add additional information during your education of biochemistry, to the end of your education, to the final exam. You will use this scheme all the time, by the way. Actually, ORO is one topic from your syllabus. And let's start from the beginning. I will start to explain first the first phase with carbohydrates, which are here on the left. But before that, let's talking about the digestion before that, to see that these processes are analogous in our organism. During the digestion, we start from carbohydrate food. After that, thanks to alpha amylase in the mouth, and pancreatic amylase in the small intestines, all carbohydrates are digested to monosaccharides and some other enzymes, monosaccharides. These monosaccharides are primarily glucose, but also fructose and galactose. And that's all about the digestion. Absolutely similar digestion, but it's not called digestion, it is called with other name, there is in our cells, because we have carbohydrate in the cells. And this carbohydrate, which is storage carbohydrate, is called glycogen. Glycogen is written here, and the blue arrows in our scheme means catabolic processes. The red arrows, anabolic ones. The process from glycogen to glucose is called glycogenolysis. You have to know the processes names. Glycogenolysis means from glycogen to glucose. And the main enzyme of glycogenolysis, yeah, actually you know from the previous lectures, is called glycogen phosphorylase. GP means this, and P means that the active form is its phosphorylated form. One more information that you know until now is that glucagon and adrenaline activate this enzyme, which means glucagon and adrenaline activate the glycogenolysis. The opposite reaction. The opposite reaction from glucose to glycogen. The process is called glycogenogenesis or glycogen synthesis, will be better to understand. And the main enzyme is called glycogen synthase that you know from the previous lectures. This enzyme is active in its dephosphorylated form and insulin activates it. That's all about the first phase of glycogen metabolism. Let's talk about proteins in the first phase. The proteins in the first phase, let's start again from the digestion. After the protein food, the digestion starts where, in which organ, do you know? You know, but do you remember? Yes? No, in the stomach. So, yes, in the stomach by the enzyme pepsin. Yes, pepsin. This is the place of the beginning of the protein digestion, and in the small intestine all proteins are digested to separate amino acids. Yes, that's why if we talk about intake of collagen or any protein cannot be resolved absorbed in the blood direct, cannot, because all these are proteins. They will be digested to separate amino acid in the small intestine, and just separate amino acid will go to the blood. That's all of protein digestion. We have degradation of proteins in our body too, because we have proteins. But remember, students, we don't have protein storage. No, we don't have protein storage like glycogen. We have different proteins in our body, and you know from the first lecture they have many different functions, receptors, hormones, they can be enzymes, and etc. After this process from proteins to amino acids, which are the end products, they are produced, the process name is proteolysis. Proteolysis. This process is activated by cortisol, by contrainsular hormones. Do you remember what I said about the contrainsular hormones? Glucagon, adrenaline, cortisol, etc. What kind of state we have during this time? Well-fed state or starvation state? Starvation state, students. If there is proteolysis and oolysis, the state is starvation state. And the opposite processes from amino acids to proteins, of course we have these processes, and they are activated by insulin. And one more, only here the information comes from DNA. Remember that. Only here, no any other compounds can be synthesized from DNA, only proteins. This is called endogenous synthesis of proteins. When we're talking about the proteolysis, it is regulatory mechanism, strictly regulatory mechanism. Actually, you know something about that, because we talked something about ubiquitination. Do you remember? Ubiquitin can be attached to old proteins and can be directed for the degradation in proteasomes. This is called proteolysis. And that's all about proteins. Let's talk about lipids. Lipid, students, are very big classification. One of them is triacylglycerols, TAG. The degradation of TAG to fatty acids and glycerol is called lipolysis or lipolysis. This is the degradation of TAG. This process is catalyzed by the main enzyme hormone-sensitive lipase. You know this enzyme, again, from the previous lectures. This enzyme is active in its phosphorylated form and is activated by contrainsular hormones, glucagon, adrenaline. And one more, which state will be if the lipolysis is active? Well-fed state or starvation state? The starvation state, yes, because the glucagon is active during this time. The opposite process from fatty acids plus glycerol to TAG is activated by insulin, and process is called TAG synthesis, but not lipogenesis. It's not called lipogenesis. And that's all about the degradation of lipids in first phase. Let's start talking about the second phase to understand the processes names. Glucose undergoes process of glycolysis to the end product pyruvate. This process is called glycolysis. It is a degradation of glucose to pyruvate. Pyruvate can go to lactate by lactate dehydrogenase enzyme with cofactor NADH, but only under anaerobic conditions, or under aerobic conditions can enter the mitochondria and undergoes process of oxidative decarboxylation by catalyzed by PDH complex that you know too. This complex is pyruvate dehydrogenase complex, and it is active in dephosphorylated form. The end product is acetyl coenzyme A, and the process occurs in mitochondria. But the glycolysis occurs where, students? In the cytosol, yes. What is produced during the glycolysis? 2 ATP at substrate level and NADH is produced. Let's see the metabolic fate of NADH. The first direction is under anaerobic conditions to be used as a cofactor of LDH. You know this reaction again. The second model is to enter the mitochondria via two shuttles that you know. Malate shuttle and glycerol triphosphate shuttle and NADH from glycolysis can enter the mitochondria. And that's all about catabolic processes of glucose. Anabolic, the opposite ones. There is this retarol, which starts from oxaloacetate to glucose. This process is called gluconeogenesis. Which hormones activate both of the processes? Let's see. Glycolysis is activated by insulin. Gluconeogenesis is activated by glucagon and adrenaline. What does it mean, gluconeogenesis? This means synthesis of glucose. What is this glycolysis? Degradation of glucose. Two opposite processes. These two enzymes, this enzyme that is written here is phosphofructokinase 2, which is active in dephosphorylated form from glycolysis. And there is one more, which is called pyruvate kinase. And that's all about carbohydrates, the second phase. Amino acids. The amino acid students can be degraded to pyruvate. Follow the blue arrows. Amino acids to pyruvate. Some amino acids are converted to acetyl coenzyme A, and all the rest to Krebs cycle intermediates. That's all. Opposite processes. Look, the retarols. They can be from pyruvate, from Krebs cycle intermediates, but which ones only? Only non-essential amino acids, because the essential ones cannot be synthesized in our body. We need to take them only from the food. No by supplements, only from the food. We have food. We don't need supplements. If there is no laboratory tested results that you have some absolutely deficiencies only in this situation. And let's see the lipids. The blue arrow, we start with degradation of fatty acids. Fatty acid degradation is called, look students, beta oxidation. This is the name of fatty acid degradation. Fatty acid degradation, beta oxidation. Glucose degradation, glycolysis. These are names. You have to know these names. Where does it occur, beta oxidation? Where, students? In mitochondria. What is produced during the beta oxidation? One NADH, which goes directly to the first complex in the respiratory chain, and one FAGH, which goes directly to the coenzyme Q in the respiratory chain. That's all. Which hormone activates beta oxidation? It is a glucagon, adrenaline, contrainsular hormones. The opposite process is called fatty acid synthesis. The substrate, acetyl coenzyme A. The product, fatty acid. Sometimes you confuse and you tell fatty acid and glycerol. No, glycerol is used only the first phase. It is used and it is produced only the first phase, not in the second phase. Fatty acids are the end products. Process is called fatty acid synthesis and also lipogenesis, by the way. Don't confuse fatty acid synthesis with TAG synthesis. Lipogenesis is called fatty acid synthesis. The process occurs in the cytosol. NADPH is used in this reaction as cofactor of reductases, because this is the only example in this scheme with classical synthetic process with a reduction reaction. That's why here is written NADPH. And PPP, pentose phosphate pathway, is the process that produces NADPH for the cell. And that's all about the two phases in the aerobic processes. Let's revise. The first phase is degradation of polymers to monomers. The second one, degradation of all these monomers to only one molecule, acetyl coenzyme A, which means never mind what is your food, what do you eat, one molecule is produced in the end, which is acetyl coenzyme A. After that, it enters the Krebs cycle, respiratory chain, and energy is produced in the end. Which are the end products after full digestion of all molecules? The waste products are only two: carbon dioxide after the exhaled, and water, which is released via the urine. That's all from the digestion. And the next processes I will explain in the next lectures. Now let's see some characteristics of biological oxidation. First, the enzymes are called oxidoreductases because we said that the processes are oxidoreduction. Number two, electrons are transported stepwise through the chains gradually. Energy is released gradually, which allows its storage in the high energy compounds. And there are two types of redox chains, or two types of biological oxidation. Substrate level phosphorylation, the first one, which is anaerobic, and respiratory chain formation of energy, which is absolutely aerobic. These are 6 main groups of oxidoreductases according to their mechanism of action. There are different classifications. Is one of the classification of oxidoreductases. Number 1 are dehydrogenases. Number 2 are reductases. Number 3 are oxidases that use oxygen as the final acceptor, monooxygenases or hydroxylases that there is involving of one oxygen atom. Number 5 are deoxygenases or oxygenases that use 2 oxygen atoms that are involved in the substrate, and the last ones are peroxidases, which break down hydrogen peroxide or other organic peroxides. We said something about redox systems. Let's see the first one, nicotinamide redox system, students, means the oxidized form, this one, after the acceptance of 2 hydrogen atoms, which means 2 electrons plus 2 protons. Only the first one are catch up into the ring of the niacin. The second one remain alone in the environment. That's why the reduced form of NADH is written NADH plus H plus, but not NADH2, no. The second redox system is Flavian redox systems, which can be FAD or FMN. 3 forms are formed: oxidized form, full oxidized form, and full reduced form, and 1 intermediate form, which is called semiquinone form. Actually, this is the free radical. The next ones are quinone redox systems with ubiquinone or coenzyme Q10. This coenzyme Q10 has 3 forms like FAG, the full oxidized form, which is called ubiquinone, the full reduced form, which is called ubiquinol, and 1 semiquinone form, which is a free radical as the same in FAG, which produces one of the most dangerous reactive oxygen species, which is called superoxide anion. It has 1 isoprenoid tail from N means 10, which means that the number of isoprenoid residues is 10 in humans. This is the coenzyme Q, which is used in the respiratory chain for the collector of electrons, can accept electrons from the complex 1 and complex 2, and can give electrons to the complex 3. This is the action of coenzyme Q as a redox system. Other redox systems are metal ions like copper 2 plus, copper 1 plus, or iron 2 plus, iron 3 plus. They form iron-sulfur complexes in the respiratory chain. The other ones are heme-containing proteins with iron which is involved in the heme. These proteins are called cytochromes. They can change the iron from 2 plus to 3 plus, unlike in the hemoglobin and myoglobin, where it's not changing in the valence. The example is lipoic acid, which can be with oxidized form with disulfide bridge, or reduced form with two sulfhydrate groups. It is used in the pyruvate dehydrogenase complex and alpha-ketoglutarate dehydrogenase complex. And the glutathione, students, which is tripeptide from glutamate. The red one is cysteine and the green one is a glycine, and this is the reduced form of glutathione called GSH. But there is full oxidized form, which is GSSG. The role of glutathione is used as a cofactor of glutathione peroxidase enzyme together with glutathione reductase because of the recovering of this glutathione. Glutathione is cofactor of glutathione peroxidase, but during this process GSH goes to GSSG. For the recovering of GSSG to reduced form, we need one more enzyme called glutathione reductase, which uses NADPH as a cofactor. Vitamin C, you know, as ascorbic acid, which works together with vitamin E. There are many reactions that are involved in the synthesis of collagen, in noradrenaline synthesis, even creatinine synthesis too. And one more very important is to improve the absorption of non-heme iron by reducing it to iron 2+. And in the end, students, there are two types of substrates for the biological oxidation. The first ones are called substrates that do not interact directly with oxygen. These are malate, you know, succinate, you know, glyceraldehyde triphosphate, and some others that directly contact with oxygen. These are phenylalanine and homogentisate as examples. And that's all from the bioenergetics and biological oxidation. Thank you, students.